Alright team, let's dive into a topic that's absolutely fundamental. We're talking about the Krebs cycle. You might also hear it called the citric acid cycle, or even the tricarboxylic acid or TCA cycle. So many names, right? But they all refer to the same critical pathway. This cycle is a central series of chemical reactions. It's used by all aerobic organisms to release stored energy. Think of it as the main engine inside your cells, constantly working. It doesn't start from scratch though, it takes fuel processed by other pathways. This is where the major energy extraction truly begins. Now why should you, as a busy medical student, care deeply about this cycle? Because it's at the heart of cellular metabolism. Understanding it is key to grasping so much about health and disease. Many medical conditions, from rare genetic disorders to common issues, can tie back to metabolic dysfunctions. The Krebs cycle isn't just a list of molecules and enzymes to memorize. It's a critical hub connecting various metabolic pathways. If this hub malfunctions, the entire cellular system feels the impact. Knowing it thoroughly will help you diagnose problems and ultimately treat your patients better. Plus, it's super high yield for your USMLE exams. Seriously, they love asking about it. So where does this incredibly important process take place? We need to go deep inside the cell. The Krebs cycle reactions occur in the mitochondrial matrix. Remember, mitochondria are often called the powerhouses of the cell. The matrix is the innermost compartment of these organelles. This specific location is highly significant. It allows for the efficient capture of high-energy electrons. These electrons are then passed to the electron transport chain, which is located on the inner mitochondrial membrane. This proximity is crucial for smooth energy production. It's all very organized in there. The Krebs cycle doesn't operate in isolation. It's a vital link in the chain of energy production. It connects glycolysis, the pathway that breaks down glucose, to oxidative phosphorylation. Oxidative phosphorylation is where the vast majority of ATP, the cell's energy currency, is made. The Krebs cycle itself only produces a small amount of ATP directly, as GTP. Its main job is to oxidize acetyl-CoA. This oxidation generates a treasure trove of high-energy electron carriers. These are molecules like NADH and FAD2. These carriers are the real prize, shuttling energy to the next stage. So how does this whole engine get revved up? The primary fuel for the Krebs cycle is a molecule called acetyl-CoA. This 2-carbon molecule is the starting point. Acetyl-CoA isn't just magically there, it has to come from somewhere. Its main precursor is pyruvate, which is the end product of glycolysis, the breakdown of glucose. Pyruvate is converted to acetyl-CoA by the pyruvate dehydrogenase complex. But that's not the only source. Acetyl-CoA can also be generated from the breakdown of fatty acids through beta oxidation. Even some amino acids can be converted into acetyl-CoA or other cycle intermediates. The very first step of the cycle is a big one. Acetyl-CoA, with its 2 carbon atoms, combines with a 4-carbon molecule called oxaloacetate. This condensation reaction forms a 6-carbon molecule named citrate. This is why the cycle is often called the citric acid cycle. The enzyme that catalyzes this crucial reaction is citrate synthase. This step is highly regulated and essentially irreversible under cellular conditions. It's the gateway into the cycle. Once citrate is formed, the cycle is officially underway, committing those carbons to the pathway. It's a major control point. Citrate then undergoes a series of transformations. First, it's isomerized into isocitrate, another 6-carbon molecule. This is just a rearrangement to prepare it for the next step. Then things get more exciting. Isocitrate is oxidized and decarboxylated, meaning it loses a molecule of carbon dioxide. This reaction produces alpha-ketoglutarate, a 5-carbon molecule. During this conversion, NAD+ is reduced to NADH, capturing high-energy electrons. The enzyme responsible is isocitrate dehydrogenase, another key regulatory point in the cycle. This is the first carbon dioxide and first NADH produced in the cycle proper. The journey continues with alpha-ketoglutarate. It also undergoes oxidative decarboxylation, losing another carbon dioxide molecule. This reaction forms succinyl-CoA, a 4-carbon molecule, and generates another molecule of NADH. The enzyme here is the alpha-ketoglutarate dehydrogenase complex, which is structurally similar to the pyruvate dehydrogenase complex. Next, succinyl-CoA is converted to succinate. This step is unique because it directly produces GTP or guanosine triphosphate through substrate-level phosphorylation. GTP can then be easily converted to ATP. Succinate is then oxidized to fumarate, producing FAD2. Fumarate is hydrated to malate, Finally, malate is oxidized back to oxaloacetate, generating a third NADH, and boom, oxaloacetate is ready to accept another acetyl-CoA, starting the cycle all over again. Your body is incredibly efficient. It doesn't just let the Krebs cycle run wild. That would be like leaving your car engine revving at full speed all the time. A massive waste of fuel and resources. So the Krebs cycle is very tightly regulated. This meticulous control ensures that the rate of the cycle matches the cell's energy demands precisely. When the cell has plenty of energy, the cycle slows down. When the cell needs more energy, like during exercise, the cycle speeds up. This regulation is critical for maintaining cellular health and energy balance. How does the cell achieve this fine-tuned control? It's primarily through the regulation of key enzymes at specific points in the cycle. There are three main enzymes that act as major control points. These are citrate synthase, step 1, isocitrate dehydrogenase, step 3, and the alpha-ketoglutarate dehydrogenase complex, step 4. Think of these enzymes as gatekeepers or traffic signals. They can be activated or inhibited by various molecules within the cell. These signals tell the enzymes whether to increase or decrease their activity, thereby controlling the overall flow through the cycle. So, what are these all-important signals? It largely comes down to the energy status of the cell. High levels of ATP, which signify that the cell has abundant energy, act as an inhibitor for these enzymes. It's the cell saying, okay, we're good on energy, let's slow down production. Similarly, high levels of NADH, a product of the cycle, also exert inhibitory effects. This is a classic example of feedback inhibition. Conversely, high levels of ADP or AMP, which indicate that energy stores are low, stimulate these enzymes. Substrate availability, like the amount of acetyl-CoA and oxaloacetate, also plays a significant role in regulating the cycle's pace. For your USMLE preparation, these regulatory aspects are super high yield. You absolutely need to know the key regulatory enzymes, citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. Isocitrate dehydrogenase is often considered the most important rate-limiting step within the cycle itself. Understand what activates them, such as ADP or calcium ions, and what inhibits them, like ATP, NADH, citrate, and succinyl-CoA. Also, don't forget the cofactors. The dehydrogenase complexes, including pyruvate dehydrogenase, which feeds the cycle, and alpha-ketoglutarate dehydrogenase, require several B vitamins: thiamine, that's B1, riboflavin, that's B2 in FAD, niacin, that's B3 in NAD, pantothenic acid, that's B5 in CoA, and lipoic acid. Deficiencies in these vitamins can severely impair the cycle. A helpful mnemonic for the substrates is, "Can I keep selling sex for money, officer?" That's citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. Why is all this biochemistry so critical in medicine? It boils down to one word: energy. The ATP ultimately generated thanks to the Krebs cycle powers nearly every activity in your body. We're talking muscle contraction, nerve impulse transmission, DNA replication, protein synthesis, active transport across membranes, literally everything that keeps you alive and functioning. If the Krebs cycle falters, the cell's ability to produce ATP plummets. This isn't just a minor inconvenience, it can have catastrophic consequences for cells, tissues, and the entire organism. Imagine a major city experiencing a prolonged power outage. Chaos ensues. Clinically, defects or problems within the Krebs cycle can lead to serious diseases. These conditions often manifest with severe neurological symptoms. The brain is an incredibly energy-hungry organ, consuming about 20% of the body's total energy despite its relatively small size. Therefore, it's exceptionally vulnerable to any disruption in ATP supply. Other symptoms can include debilitating muscle weakness, or myopathy, developmental delays in children, and an accumulation of lactic acid in the blood, known as lactic acidosis, as cells try to generate energy through anaerobic means. While these are often rare genetic disorders, studying them really underscores the cycle's vital importance. There are specific metabolic disorders directly linked to malfunctions in Krebs cycle enzymes. For instance, mutations in the gene encoding fumarase can cause fumarase deficiency. This is a rare autosomal recessive disorder characterized by severe neurological impairment, seizures, and poor growth. Another example involves succinate dehydrogenase. Mutations in SDH genes are not only linked to metabolic issues, but also to an increased risk of certain cancers. These include paragangliomas, tumors of the peripheral nervous system, and pheochromocytomas, tumors of the adrenal glands, as well as some kidney cancers. This highlights that Krebs cycle intermediates can also have roles in cell signaling and tumor suppression. So, let's wrap this up. What are the key takeaways for you? The future pillars of healthcare? The Krebs cycle is undeniably central to cellular metabolism. It's the main oxidative pathway that processes fuel molecules to generate high-energy electron carriers, NADH and FAD2, for massive ATP production via oxidative phosphorylation. It is meticulously regulated to align with the cell's energy needs. Knowing its steps, the molecules involved, its regulatory enzymes and its cofactors is extremely high yield for your board exams. More importantly, understanding its profound role in energy generation and its links to disease will make you a more insightful clinician. It's a beautiful, complex, and absolutely essential piece of our biological machinery. Master it, your future patients will thank you.